Ma'am, have you received the mail? Just now I have sent you the mail. Oh, okay, ma'am. Right. Okay.
మాట్లాడద్దు. Is it audible, sir? I request Dr. Brahma Srinivas. Please start this session.
Good morning, one and all. Myself, Mrs. A. Pratyusha, Assistant Professor, Department of Pharmacology, Hindu College of Pharmacy. I take the privilege of inviting you all to online National Faculty Development Program, ARISE, being organized by the Department of Pharmacology, Hindu College of Pharmacy. I request the convener of the program, Dr. D. Brahmasrinivas Rao, sir, to continue the session. Sir, over to you, sir, Dr. Brahmasrinivas Rao, sir. I request the convener of the program, Dr. D. Brahmasrinivas Rao, sir, to take over the session. Good morning, sir. I request the convener of the program, Dr. D. Srinivas sir, to continue the session. Over to Dr. Srinivas sir.
Very, very good morning to all the participants. Thank you for registering the FDP Arise. Once again, on behalf of management, staff, and students of Hindu College of Pharmacy, I heartily welcome to National Faculty Development Program Arise Recent Trends in Pharmacology. So why the title here? The title we mentioned recent trends in pharmacology. Selected the topics, guidelines in animal research by Dr. Vadivelan, JSS College of Pharmacy Ooty. Well, welcome to Dr. Vadivelan. Next, second topic is the research guidelines in clinical research. The third is the host immune response to present scenario, that is COVID-19. Second topic, clinical research, is going to be covered by one of the speakers, Dr. Anand Vijay Kumar sir, Professor Pharmacology, Department of Pharmacology, JSS Ooty. The third speaker is the host immune response, is going to be delivered by Dr. Raghu Chandrasekhar, Professor, Manipal Institute of Pharmaceutical Sciences. So why this particular title we have selected is, we know that research is very important. Without the research, the existence of the living being is highly difficult. The reason is that though we have the present drugs, because of the development of the resistance by different microorganisms, as well as some of the adverse effects shown by the present existing drugs, so that efficacy also, so that in terms of to get more potent drugs to reduce the adverse effects, that is to available more safer drugs, the research is very important. Again, this particular research mainly, once the drug is chemistry, its chemical pattern is discovered, then after formulation and development, formulation and development, whatever may be the drug, that must be tested in animals. That is preclinical research. Once it is successful, it must enter into the clinical phase. So both phases are research steps are very, very important in the drug development. Without a drug prove successful in animals, that cannot be tested in the humans. Here our speaker is going to cover by the guidelines. At the same time, we know that the Prevention of Cruelty to Animals Act is there for our country. Then CPCSCA, Committee for the Purpose of Control and Supervision on Experimental Animals. So that they give the guidelines from time to time in order to conduct the test on the animals. Here mainly the animals which cannot tell us any pain or suffering, so that it is our responsibility. To make less pain to the animals, at the same time safe use of the animals. Next, coming to the once it is the dose is fixed and efficacy is proved in the animals, the toxic studies are going to be done on the animals. For all this, one must follow the guidelines of the CPCA for our country. Next coming to the clinical research. Here the drug is going to be tested on the humans. The purpose of the ethics, following ethics for humans as well as animals is the safeguarding of the animals as well as safeguarding of the patients and human volunteers. Especially the humans, the human rights is very important. To safeguard the human rights of the patient or volunteer, whomever it may be, we should inform about the drugs, its adverse effects and everything, its possible drawbacks to the patient or human volunteer. Then only after that we should take the concern. At the same time, get the ethics committee approval. After sending the protocol to the ethics committee, they will see the procedures and everything. Then, if they feel the human rights are protected, at the same time the measures are taken by the investigator, then only they will give the permission to conduct the trials, to conduct the studies on the humans. So that we know that the studies are 4 trials: phase 1, phase 2, phase 3, and phase 4 trials will be there. Phase 1 and phase 1, phase 2, phase 3 trials are conducted under the controlled environment. That means first, phase 1 means unhealthy human volunteers, few members. After that, they will go to test on the patient itself. Then the third phase, different research centers. Then after that, once it is approved, then they will get the permission to market the drug. After marketing of the drug also, till the life of the drug, till the life of the drug in the market, pharmacovigilance will be there. Pharmacovigilance will be there. Continuous monitoring of the drug effects and everything from time to time, reported by the different practitioners, the physicians, as well as the other healthcare team, even the patients itself. Anyone can report. So that these are the things. These are the things in the research. Next, coming to the third phase, third one is the immune responsibility to what is that? COVID-19. We know that immunity is essential. Immunity is essential for every human being in order to fight against the infection. So that, however, to develop the drugs, a complete knowledge regarding the mechanism of action as well as how the body is going to interact with the drug. If you know, then only a successful drug can be discovered. For that sake, here we have taken the present scenario COVID-19, how it is going to react with the immune system. Then till now, what is that? Complete data we don't have. However, the scientists from different countries all over the world is going to find out how it is going to affect the COVID-19 is going to affect the human response, immune response. If it is concluded, find out, then only definitely a successful drug even will come out on COVID-19 also. So that this is the these are the some of the points why we have selected this particular FDP. So I hope all our participants learn all these things and gain the knowledge and apply it to practice. So that if you because faculty, if you know perfectly well, then you can communicate to the students who are the learners now. So once it is over, then they will follow the ethics in preclinical research, that is animal research, clinical research, and what about this other research programs. I thank you one and all. Now I welcome our dynamic chairman, Sri Jupudi Rangaraj Garu, to give his blessings and message. Welcome to our chairman, Sri Jupudi Rangaraj Garu.
Unmute. Namaste to all the members who are participating in today's FTP program. I feel it a pleasure that our Hindu College of Pharmacy has taken up this job of conducting this good program. Yesterday itself, we have seen in the papers that some medicine has been found by a company in Bombay in respect to this COVID-19. And the government of India has also given a sort of green signal. And another company in Hyderabad is trying to make another vaccine, and that's in the pipeline. At this juncture, we cannot say whether there is authenticity of the medicine for which we have to control and contain the virus. But this program will help in generating good thinking so that the children would be benefited in future. Because I see an era of not physical education, but only virtual education. This era, I feel that technology would supersede in the part of education. But however, the direct contact of education physically would more impact than these aspects of virtual training. Irrespective of this, our Hindu College of Pharmacy, which is established in the year 1999, has been catering to the needs of the pharma education. And our institutions, I can say proudly that our Hindu College High School Council institutions are started about 160 years back, and we had some legacy. And even this pharma education, we want to be a legacy. And also see that we contribute to the general public for their good health, for their good wealth, and also with virtues of learning. The virtues of learning should go to the lowest person in the society. In that view of the matter, I feel that what our Brahma Srinivas has introduced, this topic would carry, and I feel that we would still, our management is still intends to do whatever the best possible in the world for imparting good education to the children. And in that process, we feel that our college has taken up this closed virtual meeting, this closed virtual FTP program. As we are sitting in the tight room, watching from a small device, but our brain goes hither and thither. It is with anxiety to find out a good drug, a good medicine for the benefit of the society and the benefit of the world at large. Because India believes in Vasudhaiva Kutumbam. Vasudhaiva Kutumbam means in the entire hemisphere, whoever person that might be, they must be all well. With these words, I feel that our institution is trying to do whatever the best possible. And I hope that all the participants would take note of, and the speakers who are selected are also wonderful persons with enormous knowledge. So I hope that this program will go a success. But I just want to put one aspect before my closing remarks. We are very much pained by the Chinese aggression in the Galwama region. We have lost wonderful soldiers who are fighting for our motherland. So I feel that at least this meeting should at least have some pains of seeing that our motherland does not go in the hands of the Chinese. In this connection, of course, not now, in future, my request to all the persons who are participating in this program, whether in this country or overseas, or whoever they might be, China products have to be banned. China products have to be put to a stop, not. Because it is only by that process, either Vietnam or other countries surrounding the China would also get benefited. So unless isolation, just like coronavirus is to be isolated, China has to be isolated from this aspect. So my last words are, even if they pain to someone, please think over, ponder upon it in respect of the approach of the unceremonious Chinese peoples who does not bother for democracy nor values. And with this, I hope that I have not touched the hearts of the members who are on this. But my endeavor is only to see that good education is important for the children in future generations. Thank you. Bolo Bharat Mata Ki Jai. Namaste.
Thank you, sir. Thank you for your good speech. We'll follow you, sir. Now I request our secretary, sir, Sri Mansundra Garu, Dr. Mansundra Garu, to give his message and blessings.
Sir, Brahmasundar sir. Secretaries are not in line, sir.
Okay. Now I request our principal, Dr. Govindarajan Garu, to give his message.
So, good morning, all dear participants. On behalf of Hindu College of Pharmacy and the respected managements, Jubi Rangaraj Garu, chairman of this college, and Sri Masuna Garu, secretary and correspondent, and all fellow faculty members and dear students. In this college, we warm welcome one and all for these wonderful sessions going on right now. Today we are inaugurating the under the Department of Pharmacology the webinar on current trends in pharmacology. Because the title of the webinar, it may arise. The present pandemic situations arise is important because the knowledge should be arise and implement should be arise and focus to our country will be development should be arise. So in these present situations, such kind of wonderful sessions we require to impart our knowledge. For that, I am warm welcome all the senior professors, principals, and all pharma fraternity to these junctions. Now, also I request to welcome once again for today speaker, Dr. Vadivelan, professor in Jesus College of Pharmacy, and also tomorrow and day after tomorrow speakers, Dr. Anand Vijay Kumar and Raghu Chandrasekhar Garu, because all are eminent speakers. The wonderful knowledge to enlightening the people who need to do the research in present situations. This kind of the discussions are required to make it your uplift your skills. So the skill is one of the major part because we are in home, in staying home. But knowledge, we come to your place to update the required informations and share the sessions to uplift your knowledge, make with the knowledge because today this pharmacology mainly will focus on the how the research skills to get into the life because the team what they taken it is the current trends. It means the people who want to do the research are going to do the research are already there in research. Various kind of doubts are there. Such doubts, how the ethics will be focused to regulate the system of the animal research. So the various aspects are we discussing here. In these junctions, I really congratulate Dr. Brahma Srinivas Garu to choose the such eminent personalities for the speakers and also congratulate the team of the Department of Pharmacology to organize the excellent webinar. I once again to congratulate. Thank you. And over to Dr. Brahma Srinivas.
Thank you, sir. Thank you for your nice message. Now I request our Vice Principal, Dr. Sunita, to give her message.
Touch the screen. Yeah, a very fine and pleasant morning to one and all. It gives me immense pleasure to extend a warm welcome to all the dignitaries, eminent speakers, faculty members, and my dear students for Faculty Development Program on latest trends in pharmacology. The Hindu College of Pharmacy has framed a course development program, faculty development and student training program under the chairmanship of Sri Jupuri Rangaraju Garu and stewardship of Dr. S. Madhusudan Rao Garu. I strongly believe Hindu College of Pharmacy is providing a holistic education to the future generation and playing a positive role in nation building. We reiterate our endeavors to provide series of webinars accessible to all for generating global professionals. We focus on making the transmission of knowledge from the faculty to students, highly developing the next generation teachers as well as researchers. So, creating an optimized and vibrant platform for quality in knowledge enhancement and boosting the research culture and innovative skills. The webinar will keep the young brain sharp and up to date. I congratulate Dr. Brahma Srinivas and his team for their efforts to make this program in an efficient manner. Thank you, Dr. Brahma Srinivas. I wish you good luck for all.
Thank you, madam. Thank you for your good message. Now I request our OSD, Sri P. Sitaramaya Garu, to give his message.
Okay. Andariki Namaskaram. For those foreign viewers, very good morning to all of you. Well, today I would like to congratulate our Dr. Brahma Srinivas and his colleagues for arranging a very good lecture on this topic. And the speakers are also famous. I welcome Dr. Vadivelan sir, professor at JSS University, who is going to speak on ethics in preclinical studies. And another is Dr. Anand Vijay Kumar, who is going to speak ethics to be followed in clinical trials. And the another speaker, Dr. Raghu, who is from Manipal College of Pharmacy, he is going to speak about host immunization. So all these topics are very important, especially during the COVID-19 period. I am sure you are aware of some of the companies which have come with two products. One is Heterodrex, the other is Cipla. So both these have started marketing with the permission of the government of India. But the prices they have quoted today is 6,000 for one injection. So, my dear faculty members, researchers, and students, can you think how many of us can afford 6,000 for one vial? So, ultimately, all these uh, people have to work hard to bring the price down so that it is available to be common man at a reasonable price. So, these lectures will help all the faculty members, research scholars, and students to upgrade their knowledge about the ethics in preclinical studies and clinical studies and in the development of a cheaper vaccine. And as you have heard already, our management is encouraging such seminars, and all the departments are trying to focus what are the various developments in the respective areas. So I wish to thank our management for giving me this opportunity. Thank you very much for listening to my few words. Thank you, sir. Thank you, Dr. Brahma Srinivas.
Thank you, sir. Thank you for your nice message. Now I request our faculty, Pratusha, to read out the profile of Dr. Vadivelan
Thank you, sir.
on introduction also.
It's my proud privilege to introduce our today's guest speaker, Dr. R. Vadivelan, sir. He has completed his M.Pharmacy in Pharmacology in the year 2002 from JSS College of Pharmacy, Ooty, and PhD from JSS University, Mysore in 2013. He has 18 years of teaching and research experience, and his major area of interest of research is in metabolic disorders and alternatives to animal testing. He has guided 27 MPharm students and 2 PhD scholars. He has attended several national and international conferences and holds an account of overall 66 articles in peer-reviewed journals. He received various grants from ICMR, DST, SERP, and all other major projects for conducting seminars, workshops, and for improvement of infrastructure. He is a recipient of APP Best Teacher Award in 2019 at APP 8th Annual Convention and 4th Indo-US Conference held at JSS College of Pharmacy, UT. He is a present president of Indian Pharmaceutical Association IPA Nilgiris local branch UT. He is also a life member of various organizations like IPHA, IHPA, IPA, APP, and LSA. I thank the management of Hindu College of Pharmacy and the HOD of Department of Pharmacology, Dr. Brahma Srinivas sir, for giving me this opportunity to introduce such a versatile personality like Vadivelan sir. Thank you, sir. Over to Dr. Brahma Srinivas sir.
Thank you, Pratusha. Now I request today's speaker, Dr. Vadivelan, and start his session.
First of all, good morning to dear all members of Hindu College of Pharmacy. My personal wishes to the management chairman, sir, secretary sir, principal, vice principal, and all the senior faculty of the members who has introduced me as a friend of Dr. DBS. We call him as DBS. Some 18 years back, we were colleagues. Now we are having the opportunity to share our views to Hindu College of Pharmacy on the topic of preclinical research, or you can say the animal testing. And really, I'm happy to be a part of your management as a guest lecturer to deliver my ethics in animal research, the recent updates. I'm very happy to know all the members of who were giving an introduction. They were having a thorough update knowledge of COVID-19 about the coronavirus vaccine and injection Febiflu and another drug by Heterocipla, what they've introduced, COVID-4. And Sir was speaking about the Chinese companies where in future we have tried to reduce to import or export the goods and try to use the Indian system of medicine. So definitely the Indian government, we all will try to reduce the burden from these Chinese and try to introduce our product in future. So definitely near future by bringing a good animal research work and good science. We all researchers will try to improve and fulfill your dreams also, sir. Chairman, sir. Thank you, sir. Dr. Desu, shall I start the session right now?
Yes, yes, can start.
Okay. I think you can give the share screen to me so that I can start.
Okay, okay.
Sir, so nice to see you, Dr. Vadivelan, sir.
Yeah,
I accord a genial welcome to you.
Yeah, madam.
So,
thank you very much.
Yeah, we are eagerly waiting for your session. Please proceed, sir. We have given the access to you.
Thank you, madam.
84. 84.
Okay, good morning to one and all, and respected members of Hindu College of Pharmacy and today's convener, Dr. Desabrahma Srinivas Rao, and dear participants from various parts of country and global. So today my topic where I want to share my views is related with the ethical guidelines in preclinical research, the key perspectives. So right now, I would like to discuss why it is essential for us to follow the ethical guidelines in Indian scenario and what are the current guidelines available and what is the need of this animal testing in the current scenario. So what we are seeing in the COVID-19 also. So my guest lecture topics goes with some 6 important headings where we can go with the introduction, how this preclinical research existed, what is the origin of going for the preclinical research. So we all should know what is the importance of preclinical research, how it had happened, and then coming to the importance of the 3RS concept. Coming to the importance of the 3RS concept nowadays worldwide, we are following this 3RS concept. And then I think and next the ethical guidelines, what we are following the global and the Indian scenario. And coming to India, what are the regulatory framework and the animal welfare where we are following for the Indian system as they mentioned about the CPC guidelines. And then coming to the CPC guidelines for laboratory animal facility, what are the key perspectives we are going to discuss and come with the conclusion. So I am hearing some disturbance from participant side. Allow participant to unmute or
Unmuted or mute. We have muted all the participants, sir. Sir, kindly you please raise your voice because some of the participants are unable to hear your voice. Sorry for the interruption.
No problem.
Please proceed, sir. We will take care of that to avoid all those noises.
Thank you. Okay. So to start with our introduction, so if you come the two research, the worldwide what we are following as you have mentioned. So one is the preclinical research, the other one is the clinical research. And coming to any drug development process, the 5 basic process is one. starts with the discovery and development, where either we synthesize the product or we go for with some in silico studies or the isolated compound from the natural sources. So after the hit and trial method, we go with so many number of compounds, and then we come with a minimum number of compounds where we call it as preclinical research for the purpose of the animal studies. And the third phase will be the clinical research, and then the fourth phase we will go for the FTE review, and then the fifth phase will be the post-marketing services. So coming to the second phase, preclinical research. So we are going mainly to focus. What is this preclinical research? In a simple term, a preclinical research is the study which is being done on laboratory animals, where it is being done a rational before conducting the clinical trials. So here we collect a lot of data from the laboratory animals, that is the pharmacological and toxicological effects of the drugs, where we target this product for the specific patient population. So we go with the in vitro study as well as with the in vivo study for collecting the various data. So now we have seen that COVID-19, where two drugs comes into the market by Glenmark and Hetero. So one is the Fabiflu, the other one is Covifor. So these both drugs are antiviral drugs where they have been existing previously and where the animal studies has been carried out, and these drugs have been found for in the case of the safety as well as the toxicology testing with the limit dose has been safe. So that's why these drugs have been repurposed for giving for the COVID-19 issues. Otherwise, normally it will take 15 to 20 years for a drug to be developed and coming into the market. So therefore, a preclinical research, the data available in the preclinical research is definitely a more important thing for future. So what are we going to study in the preclinical studies? So here just to mention from a laboratory animal, either we go with the in vitro test by isolated organs or by cell lines, or we go with the in vivo test. So that we call it as the efficacy test on the animals. And then we go for to know the exact mechanism of action of that particular drug. Either we may go for a toxicology testing or for a safety testing or for a potency testing. So therefore,
Sir, excuse me, sir.
hello.
Hello.
Hello. Yeah.
Sir, your voice is too low, sir.
This is too low. Just a minute. No, it is kept 100, no? Is it audible now? Hello? Hello?
Hello.
Hello? Is it audible? Hello?
Sir.
Is it clear?
No, sir.
I am getting good voice here. Hello?
Yeah. Hello.
Hello? Hello? Is it clear? Hello?
Yeah. Okay, sir. Now it's okay.
Also, right. Should we start from first?
Yes, sir.
Okay. Okay, that was the topics I said which I would like to discuss. So the first we'll start with the introduction, and then we'll go with the preclinical research, why it is important and what are the types of the preclinical research, and then the 3RS concept, the most widely used nowadays throughout the world, and then the ethical guidelines, global and Indian scenario, and then the regulatory framework and animal welfare in India, as we have mentioned the CPC guidelines for the laboratory animal facility, what are the key perspectives, and ends with conclusion. So as I mentioned to you, there are 5 different stages of the drug discovery development. So one is the discovery and development by either synthesizing a product or from the isolation from the natural source or by hit and trial method by in silico studies. So followed by this, we go with a few compounds for the preclinical research and then the clinical development and then the further process of getting it approval from FDA and post-market monitoring. So the second phase in a drug development process, we call it the preclinical research, or you call it the preclinical testing in animals, or a non-clinical research. So this is being carried out to establish the rationale for conducting the clinical trial. So whatever the data which is obtained from this preclinical research, it will be helpful, that is the pharmacological and toxicological effects of this may target the product for the specific patient population. In today's current scenario for COVID-19, without animal research, we know it is not possible. So whatever the two drugs which have come in the last couple of days into the market, that is Fabiflu and Poviflor by the brand name, that is Favipiravir and Remdesivir. So these two drugs as an antiviral drug, they have been carried, which has been used for some other purpose, where their pharmacological and toxicological and the safety profile of the drug is already been carried in the animal studies, and now they're trying to repurpose it for the COVID-19. So therefore, the preclinical research and the laboratory animal testing plays an important role in a drug discovery development process. So normally, the preclinical research, we go with the in vitro test by using the isolated organs or the cell culture. That is also from the basic source from the animals, and directly doing the in vivo study, that is the whole animal studies, to know the efficacy of the product or to know the mechanism of action, and also the toxicological test, the safety probability test. So all this being carried out. Once we have a positive results, then only it will be going into the next phase of clinical trial for IND filling. So this is the way how a preclinical research takes place. And coming to the animal ethics, it comes a major role nowadays before starting a animal testing. So this animal ethics have come stringent in Indian system from 1998 onwards, where the CPC guidelines have come forward and amended a rule. So here it examines the human-animal relationship. So that is the slide which is being shown. What is the relation between the human-animal in moral consideration of animals, the moral consideration of the animals, and how the non-human animals ought to be treated. So that is very, very important, where as they mentioned, the CPC guidelines and the Institutional Animal Ethics Committee in the Indian scenario, whenever they're giving the approval for any animal studies, they care first how the moral animal is being treated. So this moral ethics is nothing but it is a science of knowledge of philosophy, where they have some more principles, such as what rules they have framed, whether it is a right or wrong, or what is the regulations, what is the research, the moral principles, what are the ethical practices we follow, and what is the code of conduct. So then some people say it is good and some people say whether it is bad. So the justification is very, very important in the case of preclinical research. So now we always fight for these aspects. The first thing is the animal rights. Second one is the animal welfare and the animal law. So every time we see the updated law, and the animal cognition, that is the capacity of thinking of the animal to also be considered, the wildlife conservation, the moral status of non-human animals. So what respect we give to the animals, and the history of the moral use. So these are considered important while doing a preclinical animal testing. So every country, they have their own rules, regulations, or guidelines to be followed. And some of the guidelines such as FDA, ICH, OECD, they have been followed by all the member countries. So that is the picture where I have shown. Some members, they fight for animal rights, and some members fight for the animal ethics. Generally, both of them overlap. Basically, we can see some two examples. A practical pursuit applying mortality to everyday situations. So whenever we do a research in animals, we find advancement in scientific knowledge, but definitely we go for sacrificing the animals. Second, an academic field asking how we should treat the animals. The human endpoints are considered to be important. A doctrine advocating the rights of the animal. So what right is being used for the animal? See, animals will not able to sacrifice themselves. Their sacrificing power is given to the humans. So we, for our sake, we should not able to sacrifice the animals. So we should follow some ethics. So for example, we can go for euthanasia. And then we have to concentrate on sentient animals. So all the animals what we are using, we can say from lower sentience to higher sentience. That is, from invertebrates to a vertebrate. We can know the feeling of pain or distress to be considered. But ethics says it considers all animals. There is no case of sentience or there is no case of non-sentience. And then you can see, always we live in the animal holocaust. Definitely, every year worldwide, we know for the purpose of advancement of knowledge, for academic or for research. So we use plenty of animals are to be killed or to be sacrificed. So they're given in plenty of numbers. So therefore, this animal rights should be considered, animal ethics should be considered, animal welfare should be considered, and human endpoint should be considered. The severity assessment is also to be considered while performing a preclinical animal testing. Now a question is asked by the researchers. Why this preclinical research or the animal research is necessary? So put in a simple word, it is essential for the science. So definitely we have a positive thoughts and a positive science for the scientific research. We always do a research for the purpose of science and for the improvement in the scientific research where it is being benefited for all the people globally. Second one, it may be used as a model to study the human anatomy and physiology where it is being used nowadays in the academic institution from a long period of time in biomedical sciences. And a model to study a disease. Either it may be used to study a Parkinson disease or a diabetes disease. So animal model is useful. And to develop the medical treatments and also for the prevention of a disease. So what we are seeing in the COVID-19, the vaccinations. And to determine the toxicity of the medications, check the safety of the product. So checking the safety of the product also, once again, we go with the animal studies. So ultimately, this animal research is a safety and beneficial to human, animal. So that's what the human-animal relationship and also for the environment. So definitely, the human and animal both are being benefited doing a study in animal, and definitely the entire environment is benefited. So these are the few clips why we go for a animal research. So we use plenty of animals from, you can say, rodents or non-rodents, or from small animals to large laboratory animals. So for example, just to highlight few, rat and mouse, they are mainly used for cancer research, metabolic disease, and for genome research. And if you see rabbit, what we see for the vaccine development and test for the various drug activity. And you go for the pig, it is used for the various activities such as transplantation, diabetes, cardiovascular disease, osteoporosis. Cow and horse, they are also used for the vaccine development, mainly in the veterinary research. And dog is used for, mainly the beagle dogs. They are used for the cardiovascular research. And cat, they are used for the cardiovascular, mainly the heart surgery and neuropsychological studies. So plenty of animals are being used throughout the world for the research and for the beneficial of the human, animal, and the environment. So we have to follow some ethics and guidelines also. So coming to the history of animal research, so we can see that the vivisection, that you call the dissection of live animals, were written in a book circa 500 BC. And so many scientists such as Aristotle and Aristotle, so these scientists, they already worked with the animals and they discovered the functions of the living organisms. And the scientist Theophrastus, so he disagreed for objecting to the vivisection of animals. So previously the physician thought that animals are less sentient. So discovering the function of living organisms or doing studies with animals is not reliable. We may not get a good discovery or a function. But this scientist, he fought for that and he disagreed the term. So here you can see a dog vivisection which is being painted in 18th century itself, where they were doing the vivisection and it is painted by a scientist. That means in 1832 itself, they have been using a dog for the experimental purpose. And then coming to Galen, the Roman physician in the 130-200 AD, he followed the practice of animal vivisection with his students, and he developed plenty of theories in physiology, pathology, and pharmacology. Another scientist, you can see French philosopher René Descartes, he coined the term automata, where the animal also do not feel the pain. And if you see, previously they thought that the circulation of the blood is taking place only through the lungs. And this scientist, the English physician William Harvey, he discovered that that is the heart where circulation takes place and not the lungs, which is the blood is circulated through the body. So like that, in the 18th and early 19th centuries, slowly they started the animal testing. And simultaneously, another thing which you call the anti-vivisection committee and ethical guidelines were also being followed. You can see the French physiologist Claude Bernard and his philosopher, you can see student Oswald de Schimmeldeberg, who is called the father of modern pharmacology. So they have introduced to the study of experimental medicine and what is the ethics to be followed and what is the benefit of the ethics to the animal welfare. And in Great Britain, Queen Victoria had an anti-vivisection campaign where it is related with the Cruelty to the Animal Act of 1876. So the history of animal testing in vivisection and anti-vivisection goes like that. And all pharmacologists will not forget this conditional reflex by training the dog by a buzzer or the alarm by keeping the food, that is stimulating the saliva, is Evan Pavlov. And in 1959, I think we all know that the Principles of Human Experimental Techniques, a book written by a zoologist, where they have coined the word 3Rs, professor and a student, that is William Russell and a microbiologist Rex Boch. So they coined the word 3Rs, where we are going to learn what is the importance of 3Rs in the current scenario of the animal testing. So coming to the types of preclinical studies, so plenty of studies are being carried. So I highlighted the few types of preclinical studies before going to the ethical guidelines. So the first study, we call it as a simple screening test, which is being carried to know the pharmacodynamic activity. For example, you may be taking a new drug and it may be done a simple screening test to know the new drug may be having a analgesic activity. So that is the pharmacodynamic activity. It is called a simple screening test for a new drug. And another type of study where we go with the isolated organs and bacterial cultures. So test on done on that. It may be useful to know the antibacterial activity or antihistaminic activity or a vasodilation activity. So by the isolated organ bath setup, lag and drop setups, we can study. And then test on animal models. So we are having a specific disease model. So we can test directly, we know the Vogel's drug discovery and evaluation book. they have a plenty of models been mentioned, where some models have been updated also. So we initially start with the smaller animals such as mouse and rat, and once getting a satisfaction result, we may go with the higher species with dogs, pigs, or monkeys. So that is one type of test. Another is the general observation test or the functional observation test, where we normally give a tripling dose to a mouse and to see all some 24 to 25 parameters. You call it the FOB test, where we check all the parameters for the drug in a smaller animal. Next comes the confirmatory test and analog activities. So first we would like to know what test is desirable to conduct, for example, analgesic activity and an anti-inflammatory activity test. And then it may be carried in a complex test in order to confirm along with this analog activity. So that test may vary from desirable to a complex. And another test where we'll be able to go for mechanism of action specifically. So nowadays, whatever the test we do, finally they ask what might be the mechanism of the drug, what is your proposed hypothesis where the drug may act. For example, you take an antihypertensive drug. So the various mechanisms are there. For example, by angiotensin converting mechanism enzyme, or AT converting, or calcium channel blocker, or beta blockers. So we may target a specific calcium channel blocker activity and we may compare. Another test is systemic pharmacology, where we are targeting the specific organs to see any defect in the organs, any efficacy, such as nervous system or cardiovascular system. And we go with the quantitative test also. For example, we may go with the dose response curve. We may know already existing drug which is available as per the IP or the BP, the range. So we may take a new drug, and the new drug is being compared with the effect of the already available drug. So we normally what we carry it with the isolation tissues from the animal. And next one is the pharmacokinetics. So we also are able to know mainly the rabbit is being used or the rat is being used for carrying out the absorption, distribution, metabolism and excretion studies, where it will be able to assess the route of administration. So that may be a beneficial type of study also. And finally, another study of toxicity testing. So where we follow the OECD guidelines also, where it may be carried with a short term or a long term. So we use the term acute toxicity study, subacute toxicity study, subchronic and chronic toxicity study. So all these toxicity studies will be based upon the duration and the number of the animals which is being used. Another most important thing to follow in the ethics and the animal welfare, where it is been developed by the Britain's Farm Animal Welfare Council in 1965, 5 freedoms of animal welfare. What are the 5 freedoms of animal welfare? Freedom from hunger and thirst. So we used to provide the animal with the proper diet and the water daily in the animal house. The second one is the freedom from discomfort. So the living condition of the animal to be considered. A proper living condition, the animal husbandry should be given. And freedom from pain, injury or distress. So normally we only observe the pain, injury from an animal. The proper diagnosis and test should be confirmed to know what may be the reason for the pain and the injury. And the fourth freedom is freedom to express from normal behavior. So here what happens, we have to give enough environment for the animal to move and to express his behavior. That is the fourth freedom. And the fifth freedom is from fear and distress. So this fear, pain and distress, this is the motto of CPC guidelines, where they are performing nowadays and mentioned in the guidelines. So a person who is following the ethics and animal welfare, he should always follow these 5 freedoms of animal welfare. And next, coming to the dilemma of saving an animal or human. So many animal research ethics questions are asked nowadays. First one, what are the substantial benefits which are associated for the research involving animals? Yes, what are the substantial benefits? Why should the use of animals require that special justification? So first thing, the benefit should be given importance. Second, while we are using the animals, it should be justified properly. What is the need of using the animals in the research? As I mentioned to you, is there any new drug discovery for the advancement of science, or whether it is prolonging the life of the human? And third one, what are the specific issues need to be considered in context of research? Yes. Then it comes about the alternatives. Before going to the animal study, what is the role of alternatives, or whether the unavailability of the alternatives play in a justification of research involving the animals? Finally, we should be able to justify the animal research relate to the justification of other uses such as food production also. And next, finally, while we are performing this animal research, other we are following a proper rules and regulations for the research involving the animals. So therefore, these things to be considered, and plenty of ethical research questions still under the dilemma. And what are some of the moral relevant features and factors? So some 5 relevant features. The sentience is to be considered. So what is sentience? So the capacity to feel pleasure and pain. The capacity to feel pleasure and pain. The humans feel that, even the animals also feel that, so a proper care should be taken. Second one is the higher cognitive capacities. So that is the ability to use the language, and you know that it can have the learn to the complete tasks such as making and using tools. So the brain. So that is also to be considered. The capacity to flourish, that feature says the ability to satisfy species specific needs. The capacity to satisfy species specific needs. And the other relevant feature is sociability. So being a member of the community, what is the social responsibility in handling the animals in the research? Next one is the possession of life. So definitely attributing value to life itself. Attributing value to life itself. So these are some of the relevant features. And coming to factors, what are the goals of research? So the same points have been discussed here. What are we going to achieve? That is the goals of research. What is the probability of success? Either it is a positive or the negative. Which animals are to be used? Are we going for a lower animals, lower phylogenetic scale, or the higher species? Then effect on the animals used. So what effect has been observed in the animals? Are there any alternatives? So these 5 factors and 5 relevant features are to be considered in an animal research. And definitely, as I mentioned, there are pros and cons. What I mentioned, animal ethics as well as animal rights. So the pros is that the good thing, solution for ethical question of testing. We have ethical question. But the cons, then a bad one, or the negative aspect says, whatever maybe, it is a painful experiment. It's a benefits for human. The con says it is an expensive procedure. We always go with a statement saying we go for animal research because the human and animals are identical. Here they say they are non-identical. It is related for the safety of the product in pros. We say animals are poor subjects. It's not comparable with that of the human. A possibility to study entire lifespan, because we know the lifespan of either a mouse or rat is very short, either for 1 year or 2 years, maximum 3 years. But it is an exempt from Animal Welfare Act. It is a sentience animal, so respect should be given. Cost effective. Animal testing is preferred over the alternatives. Yes. Animals are benefited by the procedure, as I mentioned. The cons, the other groups of people say it is a unnecessary test. So this will be giving an advantage for going for a further studies. But they say it may not be reliable. So always there is a dilemma in the animal research. They have the pros and cons yet. And next, coming to the how this animal testing has been originated, some three tragedies alone I'll be explaining in a short way. The one is the Lashlier case. If you see in 1930 in US, an untested eyelash dye containing phenylalanine diamine, that is aniline, where it is being used for as an eyelash, Lashlier as the brand name. So what had happened? It has caused some ulcer or redness in the eye, and it has caused some blindness, and more number of people caused blindness, and at least there was one mortality. So it was not tested in the animals for the irritant test or a corrosive test, where it is being directly taken with the human as a volunteer. So here it is a need for going for a animal testing. And then they brought in US federal and cosmetic drug acts were introduced in 1938 that it should be carried out in animals. Another important thing is the elixir tragedy. So the same in US in 1937, an elixir of sulfanilamide. You know that sulfanilamide is a wonderful antibiotic where it is being given for all the bacterial infections. So they want to bring it in a form of an elixir where they use a solvent as ethylene glycol. So this tragedy, what happened? More than 170 people died throughout the US, where this ethylene glycol, which is been after dissolving sulfanilamide, it has been converted into oxalic acid. This has formed the oxalate crystals in kidney, and it has caused some toxicity in the kidney. So that is the reason for this death of the people, and that is the elixir tragedy. And other most familiar tragedy, which was coming to the extent where it has been mandatory to go with animal testing, that is the thalidomide tragedy. Nobody will be forget it. And the phocomelia is the condition where the baby were born with congenital abnormality with sealed limbs. In 1957, by a German pharmaceutical company, Grunenthal, as a risk-free sedative designed to combat the morning sickness. So normally pregnant women, so they have the morning sickness. So it is being used for that purpose as a sedative and hypnotic agent. You can see in 1961, more than 10,000 people were deformed with this congenital abnormality, that is the term phocomelia, were born in Europe, mainly in Germany. So these were the last person who were affected with this congenital abnormality, phocomelia, Louis Medes. Still they are alive. So therefore, these three tragedy, Lashlier case, sulfanilamide elixir case, and thalidomide for the origin predictive animal testing made the members in the society and the scientists, Russell and Bruch, they brought the reason for this, mainly this phocomelia tragedy, thalidomide tragedy, and they said it is a mandatory to go with the animal testing. So they coined the term as 3Rs in their book, which has been written, as I told, the principles of human experimental techniques. So we have not stopped using the animals, we have continued but modified the use of animals. So in their book, they have quoted a term, if animals were to be used in experiments, every effort should be made to replace them with non-sentient alternatives. So for making you to understand, sentience means the feeling of pain and distress, the pleasure. Non-sentience, they do not have the feeling of pleasure of pain. So we know that we and the animal having the capacity to feel the pleasure and pain. So that's what, as I said, one relevant feature, the first feature was sentience to be considered, the moral sentience. So the 3Rs is important. In the what you call the first R, always the order of these 3Rs are important in animal testing. The first one is the replacements, methods which avoid or replace the use of animals. Second R is the methods, that is reduction, methods which minimize the number of animals used for experiment. And third R is the refinement, methods which minimize the suffering and improve animal welfare. The replacement, reduction, and refinement. Now, I think the CPC guidelines, they have coined the fourth R, that is the rehabilitation, or sometimes you call it as the responsibility. The first one, what is the first R? Replacement. As I mentioned to you, substitution of insentient material in the place of the conscious higher animals, and replace the higher animals with lower animals. So, for example, a study can be done in mouse. The same study can be done with a rabbit. We go prefer with the mouse. And for the industry and the institutions, for us academic and institutions, replace live animals with dummies for teaching and dissection purpose. Use computer simulation and in vitro methods. What we are following the curriculum for experimental pharmacology, where we call the call techniques and the cell culture all are being used nowadays. The second are reduction. So this is always performed with pilot study, either with 1 or 2 or 3 animals. So that is the OECD guidelines. They made alternative to the animal toxicity testing. We follow this oral toxicity testing such as 423, 420 or 425. And design study to use animals as their own controls. We call the crossover study, and try to go for gather data for more than one experiment concurrently. And always they say consult with a statistician to go with the number of animals to be used, and minimize the variables such as disease, diet, stress, and genetics. And always we have to use appropriate species for the animal studies. So, for example, we all know that rat may not be having vomiting symptoms, so it cannot be used for an antiemetic study. So, like that, we have a rabbit may be ideal for a pharmacokinetic study, where we go for collecting of the blood with the various intervals. And finally, the refinement. That is very important. There it fights with the animal ethics and the animal welfare. The husbandry needs as per the species requirement. The animal husbandry, there are some guidelines as per the CPCCA, we'll follow. Refine experimental procedures wherever possible. And refining the management of pain. We have to go with proper sedatives or analgesics. And refining humane endpoints. So this humane endpoint is considered to be important. So we know when the animal is going to suffer, or when the animal is going to suffer from the mortality, the precaution should be taken, and we should not allow the animal to die by itself. Based on the severity assessment, we should go for euthanizing the animal. So that is about the refinement. So if we see about the 3RS, the National Center for the 3RS in UK, where there are so many member countries, they conduct, give many guidance, they host symposium and workshop, they conduct some training courses, they award some grants, and they have innovations, and they give the student fellowships also in this 3RS. Now we can see recently in last couple of days, this NC3R and DHF, that is the British Heart Foundation together, they have introduced a student fellowship for advancing cardiovascular science with 3RS methods. Some 3 methods where they are going to follow, that is with the abdominal aneurysms in human umbilical arteries, or for the in vitro occlusion clot reformation. So it is a PhD student fellowship. Interested students can appear for it. There are some guidance to be followed. And coming to the ethical research guidelines, the global scenario, the way there are different guidelines developed throughout the globe, and what are the variety of reasons. Simple thing is that we can easily compare with one compound with another compound, and that gives one species with another species. So therefore, the results of the various substances can be easily compared. And another important thing, if somebody they have done it, then we no need to repeat it. So this may encourage the use of certain protocols for testing need not to be repeated. and this may facilitate the work of those who decide and their testing guidelines. So you know that worldwide, we always remember while teaching the pharmacology students, and still we remember in the drug discovery process, I said you the FDA guidelines involving the whole animal testing, and the most commonly used guideline for testing of chemicals, mainly for the toxicity, is the OECD guidelines. So let me see in the next slide what is the OECD guidelines. And all these guidelines is not belongs to one country, where there are number of countries there following it. And normally the GLP, where we go do research for some other countries, there it is essential whether they ask whether this guideline is followed, whether your lab is with this GLP. And another one is the pesticide assessment guidelines involving the whole animal testing. So some of the guidelines, however, we are going to discuss beautifully about CPCSCA, that is the Committee for the Purpose of Control and Supervision and Experiment on Animals, where in India we are following it. Any animal house in India or a research institute, the prior thing they have to follow this guideline, rather than other testing guidelines also. The other one is the CARE, that is the Community on Animal Research and Ethics, that is in the Washington, D.C. And another guideline for care and use of animals in scientific research by India. In Singapore, they are following the National Advisory Committee for Laboratory Animal Research. And they have different countries. In Europe, they follow the different guidelines. And in America, we are having the American Psychological Association. They also follow some guidelines. And the NHMRC, National Health and Medical Research Council in Australia. So these are the few guidelines have been highlighted like that. Every country, they have an individual guideline by the veterinarian committee where it should be followed throughout their research system, animal research system. So just to highlight what are the regulatory guidelines for conducting toxicity studies. As I mentioned, in animal testing, toxicity studies also one type of the study. So one is the OECD, that is the Organization for Economic Cooperation and Development. Plenty of guidelines are available from acute, subacute, chronic, reproductive, genotoxicity, reproductive studies. Plenty of guidelines with updated are available mainly for testing on chemicals. And the second one may be the International Conference on Harmonization. Mainly they follow the QSCM, that is the quality, safety, efficacy, and the multidisciplinary, followed by ICH. This is also worldwide accepted guideline. And other is related to the Environmental Protection Agency by USA. And India, we are following the Schedule Y. The regulator is the CDSCO, mainly for Central Drug Standard Control Organization, where they have enforced under the Drugs and Cosmetic Act 1945. But the most widely used guideline for toxicity and the safety is OECD and ICH. In India, we have to follow the CPC guidelines also. So how these CPC guidelines have been evolved? To highlight, we know that about the Prevention of Cruelty to Animals, PC Act, which was initiated in 1960 and amended in 1982. The CPCSA Act, that is, was followed in the year 1964 itself. However, they brought with a new amendment of rule in 1998. Then other law, that is in 1972 by the Wildlife Protection Act. In 1992, INSA, that is the Indian National Science Academy, guidelines for catch and use of animals in scientific research, and it has been revised in 2001. And I told the important rule which was amended by CPCSA is in 1998 in today's government, that is breeding of and experiments on animals. That is control and supervision rule, which was amended in 1991, 2001, and 2006. And then they are brought by ICMR guidelines in 2001. MCI, they made the amendment for the use of animals by replacing animal experiments. In 2012, Ministry of Health and Family Welfare, they said ban use of animals in educational institutes. And in 2013, UGC guidelines for discontinuation of dissection and animal experimentation in zoology licenses. So what our institutes, the academic institutes, we follow by them. And in 2015, PCA amended that recommends to use alternatives to replace animal experiments for the academic, not for the research. So that's what we are following nowadays in the academic institutions. Next, coming mainly to our topic, the key perspectives, CPCA guidelines for laboratory animal facility. So to have updated guidelines, we can just go into the website. From there, my disclaimer, as I said, I have just taken the guideline and I have highlighted some few points. So here, if you see, previously this CPCA purpose and control supervision animal and expenditure was under the Ministry of Environment, Forest and Climatic Changes. So 4 to 5 months back, it comes under the Ministry of Fisheries, Animal Husbandry, Dairy and Veterinary Science. So it comes in a different thing, and where the veterinary science, they have taken a part of it. In that, if you're going to the guidelines, we are having a compendium of CPCCA, updated guideline in 2018. So always when seeing the guideline or the form to be filled, we have to follow the updated guidelines. So this is the updated guideline where we have taken, it is the compendium of CPCCA 2018. In here, if you see, there are 29 items. We have taken the 7th item to be discussed. CPC guidelines for laboratory animal facility, page 61 to 96. So coming to the introduction, background and aim of this guideline, as I mentioned to you, why this PCA Act in 1960 and 1982 amended is to prevent infliction of unnecessary pain or suffering to the animals. So that is their main motto, to prevent the infliction of unnecessary pain or suffering to the animals. So by that, the CPCA is framed, they should not subject unnecessary pain or suffering before, during, or after the performance of experiments on them. There are 3 things: before, during, or after the experiment. We should not subject the animals to the unnecessary pain or suffering. Care should be taken. So that's why the rule I have told you, the breeding of and experimental animals control and supervision rules was amended and it has been updated. So the main is to eliminate the potential for conflict. It was considered necessary to examine the international norms regarding the use of the animals. And most important thing is that why we follow the international norms, to update the regulations, streamline and simplify the procedures while ensuring the ethical use of animals. So the main aim of the CPC guideline is to ensure humane and ethical treatment of animals. To ensure humane and ethical treatment of animals. So what are the main functions of CPC-SCA? Registration of establishments for conducting animal experimentation or breeding of animals for this purpose. So any registration of animal facility in India, which should be registered and licensed by CPC-SCA. So they give the selection and appointment of nominees in the IAEC. So they appoint 3 nominees. One is the CPC main nominee, one is the link nominee, and the other one is the socially aware nominee, and one more is the scientist in charge from the outside facility. The remaining 4 to 5 members may be appointed from the management or the institute. And they also give the approval for the animal house facility on the basis of reports of inspection conducted by CPC-SCA, permission for conducting experiments involving use of animals. The small animals, the IAC can give the approval, but however, when going for a larger animals, they can only recommend and it will be scrutinized by the main CPC office, where it comprises of more than 25 members. And also it's helpful for recommendation for import of animals for use in the animals and action against establishment in case of violation of any legal norm and stipulation. So these are the functions of CPC-SCA. And coming to the 5 important principles of the CPC-SCA for the use of animal in scientific experiments. The nominees mainly they took care of these 5 principles before giving the approval for a protocol. First principle says experiment on animals may be carried out for the purpose of only advancement by a new discovery of physiological knowledge, or whether it is useful for saving or prolonging the human life or alleviating suffering or any significant gains in well-being of the people of country or for combating any disease. The experiments on animals should be carried only for the substantial benefits of the people and the society. Second principle, I have already told the word sentience. We have the capacity to pleasure and pain, feel. Animal of lowest on phylogenetic scale with the least degree of sentience, which may give scientifically valid disease with the use. And I show the image how the lowest phylogenetic scale starts with invertebrates to vertebrates. For example, it may start with Drosophila zebrafish, then comes the mouse and higher animals such as human. And third principle says proper use of animals in experiments and avoidance or minimization of pain and suffering inflicted on the animal. In such a case, to minimize the pain, we have to go with the appropriate sedation, analgesia, or some good analgesia can be used. Anesthesia also. And principle 4, that is the persons engaged in animal experimentation should have a moral responsibility for the welfare of animals after their use in experiments. And the investigators, the principal investigators, the veterinarians are responsible for the aftercare or the rehabilitation of animals after the experimentation and may be permitted to euthanize. So the human endpoints may be considered here for euthanasia. The principle 5, it is the living condition of the animal should be appropriate for the species and contribute to their health and comfort. In a simple word, we can say, for all the biomedical purpose, it may be directed by a veterinarian or other scientist, the housing, feeding, and care for all, which should be considered. So these are the 5 ethical principles which is being quoted in the CPCA guidelines for the use of animal in scientific experiments. To summarize these 5 principles, the principle 1 is the experiment on animals, why we should perform the experiment on animals as I mentioned. The principle 2, the animals lowest on the phylogenetic scale to be considered because the feeling of pleasure and pain. Principle 3 is the proper use of the animals and avoidance or minimization of pain and suffering. So we should use the sedatives or painkillers, that is equal analgesics or anesthesia. The principle 4, the moral responsibility for the welfare of animals, the living condition of the animals should be known. So the human endpoints are to be considered and euthanasia to be given to sacrifice the animal when it is required. And principle 5 says the living conditions of the animals and as I mentioned, the support and care for all to be considered by veterinarian or any scientist in the relevant discipline. So therefore, these 5 ethical principles are to be considered before the use of animals in the scientific experiments. And next, coming to the CPC guidelines. So they mentioned different items. Starts from your goals, veterinary care, until the transport of laboratory animals. So what are the specifications given by the CPC we have to follow? And it is also based upon our research and the number of the animals to be housed. So based on that specific guidelines, animals should be built, and it is mainly based on the research and the financial of the management and how many number of the animals should be placed. So it may be a smaller animal house or it may be an animal house which may be necessary, need-based for the institution for academic and research. To start with, first from the goal. So what is the main goal of this thing is to promote the human care for the animals. And we know about the nation of our father always says that he has quoted, the greatness of the nation and its moral progress can be judged by the way its animals are treated. So therefore, the basic objective is the advancement of biological knowledge that is relevant to humans and animals. And the second, utmost care, that veterinary care. So the animal house, the animals, the comfort, the health care, all is the moral responsibility of the veterinarian. So a daily observation of animals, by Kate said, you call it as a micro environment or a macro environment of the animals and the animal house. The veterinarian is having the most responsibility by observing it, and based on the situations of the animals, a proper care or diagnosis can be given. And animal procurement. We know that these are the various types of the animals which are used, and it should be followed as per CPCSA law guidelines. It should be obtained by registered breeders, so they'll be having a license. As I told you, the beagle dog, which has been imported from abroad, so it is mainly we have to obtain from DGFT, that is the Director General for Foreign Trade. And you can see some of the animals which can be procured, or knowing this is the mouse, a rat, hamster, golden hamster, guinea pig, rabbit, cat, and this is a nude mouse. So in most of this testing, the mice is being used preferably for the toxicity testing. Then comes to the quarantine, stabilization, and separation. So whenever they bring the animals from the outside to the inside animal house facility, so these three things to be considered. The microbial status of the newly received animal should be considered. And there are some guidelines given for the hematological parameters and biochemical parameters, which is given in the annexure 1 and 2, which I kept in the last slides. And for a smaller animal, we can give a quarantine for 1 week. And for a larger animal, we can keep for 6 weeks. So it should be kept separately. And stabilization, it is based upon the physiological, psychological, and nutritional stabilization. And separation, sometimes when the animals are infected, it should be kept separately, and a separate personnel should be there available for taking care of them also. So this may prevent the interspecies disease. You know that some contagious disease may easily be transmitted, such as tuberculosis and all, within the monkeys. So to prevent this interspecies disease transmission, and also to eliminate the anxiety. Nowadays, we know that for any type of animal house, the surveillance is important through webcam, where they used to come for inspection of the meeting. We have to show the record. So that is important nowadays. So this may be helpful for diagnosis, treatment, and control of a disease, observed for signs of illness, injury, or abnormal behavior by animal house staffs. Therefore, the contagious disease animals may be isolated from the healthy animals in the colony, and it can be vaccinated. So that's the thing, it can be shown. Personal hygiene, that is the person who is working in the laboratory animal facility. Nowadays, as I told you, they made mandatory to wear face mask when you're going outside due to COVID-19. You see, that's the importance in animal house to wear the entire, to take care of the personal hygiene. So the high standards of personal cleanliness, clothing, disposable gear, such as face mask, gloves, and change the clothing as often. So here that is the thing. And no food should be taken inside. And coming to the location of animal house facility, we should have a separate building for animal house facility, or the building may be attached a separate with that of the academic institution. You can see that is at least about 50 to 60% of the total constructed area belongs to the animal house. That is your animals where it should be placed. The remaining area for the administrative areas of animal house facility. Functional areas, so this is the image which is sketches being shown. We should have various functional areas. It is based upon the separation of species or isolation of individual projects whenever necessary. So storage areas for food, bedding, administrative supervision, and showers, sinks, lockers, or toilets, for sterilization equipment, for holding soiled or cleaned equipment, repairing cages, stored waste prior to the incineration. So nowadays we know that we are not going for incineration due to the air pollution. We go for the biomedical waste management. And as per the CPC guidelines, they're given in the annexure 3 and 4, what are the different types of anesthetics and euthanasia should be used. Mainly the sedatives and anesthetics, we know that analgesics, they used to control pain and distress under an experiment. So whenever we're performing an experiment, we have to go with the anesthesia, either it may be injectable or it may be inhalation. And overdose of anesthesia may be termed as euthanasia, that is by considering the human endpoints, we may go for a painless killing. And transgenic and a knockout animals should be first euthanized and then disposed of as prescribed by the guideline. And always we should have a record of disposable. So normally we can say 3 to 5 times of anesthesia will be a euthanasia. And what are the various physical facilities should be available in an animal house? So the building material should be moisture proof, fire resistance, and it should be more desirable for the interior surface, including vermin and pest resistance. The corridor which is available should be wide enough to facilitate the movement of personnel as well as the equipment, and it should be kept clean. We should have enormous utilities such as water lines, drain pipes, and electrical connection. And animal room doors should be free from rust, vermin, and dust proof. Floors should be smooth, moisture proof, non-absorbent, and it should be capable of supporting racks, equipments, and stored items without becoming gouged, cracked, or pitted. The drains and the physical facility, it is not essential to have a drain in all the rooms, for example, rodent rooms. Whichever is necessary, based on this facility, we can have a drain. Wall and ceiling should be free of cracks, unsealed utility penetrations, or imperfect junction with doors, ceiling, floors, and corners. And as I mentioned, separate storage areas should be designed for feed, bedding, cages, and materials not in use. Facilities for sanitizing equipment and supplies. We know that sanitizing cages, ancillary equipment is essential with adequate water supply. And experimental area where we perform on the animals, so that also should be have a separate area where the animals should be housed. And next, coming to the animal husbandry, caging, and housing system, adequate ventilation is essential and meet the biological need of the animals. It may vary from species to species. For a mouse, rat, we can have a small cage which is made up of this plastic, which is stainless grade. And in the case of the rabbit or a guinea pig or a higher animals, we may have a different types of cage. So it should meet the biological need of an animal. It should be kept, the animal should be kept clean and dry. We should facilitate research while maintaining good health of the animals. As I mentioned, the cages may be made of steel or painted whenever needed. Feeding and water facilities devices should be easily accessible for filing, changing, and cleaning and servicing. So always it should be easily accessible. Food, I think we fed the palatable or non-contaminated and nutritionally adequate food. So it is also based on the species. For mouse and rat, we can give a pellet. And in case of some animals such as the rabbit, we can go weekly once with that of the vegetables also. And we should always have a sufficient to ensure for the normal growth in immature animals. A stock should be kept, and it should be clean, and it should be free from heavy metals. And coming to the environment, the air conditioning is always acceptable, and the temperature within the range should be 64.4 to 84 Fahrenheit. The relative humidity may be around 30 to 70%. It may vary slightly for the animals. Power and lighting should be for 24 hours, and it should be available in the case of the emergency also. And noise control, that's what I said, it should be a separate building and it should be free from noise. So this can be a simple setup where it is being shown. It is recording the humidity, temperature, and the time. Bedding, once again, it is based upon the research where you perform. It should be absorbent and free of toxic chemicals. It should be easily stored, uncontaminated, non-toxic, that's what I said. and disposable by the incineration. So normally we use the paddy husk or the wood chips, which is somewhat costly, are being used. And water should be fresh, potable, and uncontaminated. Waste disposal, as I mentioned to you, nowadays it should be preferred with incineration, and the waste must be stored before removal. There should be separate storage area for flies, cockroaches, rodents. Nowadays, we go for the biomedical waste management, how they perform in the hospital. This is just maintaining incineration, and as I told, the municipality may not give a permission. So we have to use a different types of buckets, which different colors, where it can be kept, and the people, whenever you call them, they come and collect it and they will go. And we should have a pest control program also to be designed to eliminate the presence or infestation by the pest. So program should be organized. And we know there is no weekend holiday for the animal house facility. Every day they should work, and always to safeguard the well-being, emergency veterinary care should be available with the qualified personnel every day. Record keeping, every record should be kept for the animal house facility during the inspection. Animal house plan, staff record, health record of animals, all relevant SOPs, breeding stock purchase sales record, minutes of the institutional animal ethics committee, and record for the number of animals used, death record, clinical record of sick animals, and water analysis report. So these all are mandatory, and this will be verified by the nominees during the inspection. And for every facility in the animal house, what we are having, we should follow some SOPs. It can be made in a simple by the investigators or the person, the scientist in charge who take care of the animal house. So that simple we ask where, why, how, what. So such a way we should mention, including for all the equipments we are using, and it should be always updated with the signatures. The animal care and the technical person who is working in the animal house other than the veterinarian. So you should be trained with the laboratory animal and provide for both formal and for job training to ensure effective implementation of the program. So previously they have said that 10th qualified person is essential. Nowadays they mention the person should undergo a course. It is a 15 days to 1 month course where it has been adopted in Hyderabad, and also the CFTR in Mysore, where they are having 5 chapters where I have shown in the annexure 6. So it will be useful for those persons. It is related with the animals, how to handle, how to inject, and a knowledge about those animals. And finally, coming to the transport of laboratory animals is also important. Whenever you are bringing the animal from one place to another, based upon your research, the main consideration for the transport of animals are mode of transporter, either it may be air, road or rail, and the container, whatever we are using, and the capacity of the animal, that is the animal density of cages, and food and water during transit, and protection from the transit infections, injuries, and stress. So this is the thing which I shown, the mode of transport and how the animal should be kept in the cage where they have a comfortable movement and protection from possible injuries. So it should be made available for reducing the space parallelly by decreasing the time of transportation. And food and water should be adequate and more particularly fluid during the transit, and it should be appropriate size, the cage, for avoid overcrowding and infighting, where it is being shown in annexure 4. So this is one cage which is being shown where it is kept with the husk, and for the free movement of the animal and the water can be kept. So that is about the various items to be discussed regarding the CPC guidelines for the laboratory animal facility. There are some 8 annexures where I have shown here. However, I am not going to read all the annexures where you can find out through this compendium. So annexure 1 is related with the hematological data for common laboratory animals. Annexure 2 is for the biochemical data for common laboratory animals. and 3A, 3B up to 3G, it is related with the recommended space for the laboratory animals. And Annexure 4 is related with the specifications for transport of laboratory animals per road, rail, and air. And 5 is related with anesthesia, 6 with the euthanasia, and 7 with the qualification and knowledge required for a laboratory attendant. And 8 may not be a part of that, but it is related with the Institutional Biosafety Committee, IBSC. So some chemicals or some microorganisms which are involved with the hazardous. So previously they should get approval from IBSC. For example, you're going to prepare any vaccines. So for this, with the microorganisms, we should first get approval from the IBSC. Then only for the approval of animals, it comes to the IAC meeting for the approval. Some 8 annexures are there in the case of the CPC guidelines for the laboratory animal facility. So these are the annexures where it has been updated as per the 2018. So you can go through that. They're given for some 7 species. What is the range of the hematological data? What is the range of the biochemical data? Is been given, as I told you, when some animals are brought from outside to the inside animal house, which is kept for separation, quarantine, stabilization. So in order to know the microbial status of these animals, when needed, these parameters to be checked. So this is related with the space area. Okay, fine. And Annexure 4, as I mentioned, with what is the cage size, what type of cage, and how many number of animals as per the space per square centimeter to be utilized. And anesthetic agents, it is nowadays difficult to get all these anesthetic agent without prior letter from the institute, going to a medical shop. And as I mentioned, general anesthetics, we are having inhaled and as well as intravenous, what we are following. Nowadays, the most easily available is ketamine and xylazine, where you can go for combination for anesthesia. And these are the various euthanasia for laboratory animals, where we have mentioned the physical methods and inhalation and drug administration. So first, we try to follow the physical method when the weight is less than 200 grams. Or then based on the facility, we can go for drug administration, injectable. If you're having the carbon dioxide chamber or monoxide chamber, we can go, all those are acceptable methods. So these are the images where it has been followed long back for the physical methods. As I told, that is the qualification and knowledge required for laboratory attendants. Some chapters are mentioned. We should be getting trained in it. And IPSC, where we go for prior approval, the dealing with hazardous substances of microorganisms. So that's all about the topic regarding with the ethical guidelines for the preclinical research. So in my slide, I have not touched about any specific guideline to be followed either by OECD or for the efficacy study or toxicity study or reproductive study or safety study. So my motto of doing things, what are the ethical principles, what are the ethical guidelines, what is the dilemma, and what is the animal rights, animal welfare to be considered. And the key perspectives I mentioned about CPC guidelines, what all should be available and what we should follow in Indian system while we are following the animal studies, the preclinical research. For a simple research for a PG student or a PhD, or you go for a trade or marketing. So the animal house may be of 3 to 4 licenses. One is for academic, for basic research. Second one, it may be for breeding. Third one, it may be for trade. And fourth one may be for commercial. So most of the animal house we may get for the basic research and for breeding. So once we go for breeding, we should be able to maintain it. The trade and commercial, where we go for doing more research or we're going for some product. So I would like to conclude by saying for the animal welfare and rights, I can't speak for myself. You are my voice. So that's why we all scientists and academicians, we speak for the animals, for their welfare, considering the human endpoints, and don't treat animals as animals, because I told they are also sentience. They are having the capacity to feel pleasure and pain. Treat them as living beings. That's what they are there. And on behalf of the animal welfare rights and animals, I thank you all for giving me this wonderful opportunity to discuss. Uh, if any queries, I'm happy to answer to your queries related with these ethical guidelines followed in Indian society. Yeah, Desu?
Oh.
Yeah, you're audible. Yeah.
This is question number only, so that
Yeah.
wait.
You wait for.
Anil?
Sir.
Uh,
Sir.
so look into the questions in the chat box as well as arranged by the participants.
Okay, sir.
For question also.
Good morning, one and all. Good morning, sir. This is Anil from Hindu College of Pharmacy. There are three questions from Sridevi and as well as Satish Kumar. So I would like to ask from one question from Sridevi. Can you please explain what kind of testings can be performed on zebrafish? This is the question raised by Sridevi.
Okay, next.
Can you please explain what kind of testings can be performed on zebrafish?
Right. So in the case of using zebrafish, we can go for mainly the reproductive toxicity studies. We can go for doing reproductive toxicity studies in different stage, because in different stage we can go for sacrificing the animal and we can perform it. We know that. And second one, you can go with the neurotoxicity testing also. You can go with neurotoxicity testing. So mainly zebrafish, we prefer we go with the reproductive toxicity testing. The advantage is there because we can use more number of animals in the different stage.
Thank you, sir. And the next question is from the same person. How to dispose the sacrificed animals as per the ethical guidelines?
Okay, good question. And this was asked by everybody before I am giving the presentation. So you see, as per the CPC guidelines, I have shown one equipment which is called as incinerator. So this was the first, if you see some 18, 20 years back. Initially, they have said each animal house facility, we have to go with the incinerator. So once you put the animal inside there, you know that it will be converted into an ash. And what happens? This the municipality of the local town, they have made that it may cause unnecessary air pollution. So what they have done nowadays, as I told, the biomedical waste management. It may be a private company also where we should have a license from them. And I showed you in my slide different types of buckets with different types of colors have been kept. So we may not have every day that we are not exposing the sacrificing the animals. Whenever we have sacrificed and plenty of animals are there, once you call them by phone, they will come and collect it and they will go and dispose it. But it is not possible for us to bury under using a lime under the ground and all is not possible by our thing. They say follow the biomedical waste management, and when they come for inspecting the animal house also, they will ask you to go the certificate and the license.
Thank you, sir. And one more question from B. Rajesh. In 3R reduction, how many number of animals will be given significant p-values?
Yeah. So this is a very good question. Some of them they are saying if you go with three animals also, it will give significant values. While you're going with three animals, because you imagine you're taking animal, I shown one of the reduction. See, in one animal, you can go for a biochemical parameter, you can go for a hematological parameter, and you can go for histopathology. So, in those cases, we cannot use all the six animals for all the purpose. So, if n is equal to 3 also, it will give the statistics value. but as per the statistics, they say minimum of 6 animals will give a good p-value. But a good answer, how many number of the animals to be used and the statistics, a statistician will be giving a better answer. He will work out the numbers.
Yes, thank you, sir. And one more question, sir. Uh, Along with the CPCSC, is there any other norms to follow the rules and regulations for carrying out preclinical studies?
Yeah, good question. So I said the basic guideline, the basic criteria in India, in your animal house, so the CPC guidelines is related for the facility for animal house. So that's why it is highlighted. CPC guidelines for animal house facility. In animal house facility, they have followed some of the international norms and they have given some of the guidelines. But I told you, well, if you want to follow some of the guidelines, more guidelines for toxicity testing or for safety pharmacology, we can follow the guidelines from OECD or from ICH for the good laboratory animal practice. There is no wrong in it. But the animal house facility minimum in India, it should be as per the CPC guidelines. You may have a extra also. That is not an issue.
Okay, sir. Thank you, sir. Now I will hand over to Dr. Desubrahma Srinivasarao.
Very nice presentation by Dr. Vadivelan. Thank you, Vadivelan. Thank you for your nice lecture.
Thank you.
On guidelines of preclinical research. Now, I thank our chairman, Sri Jupudi Rangaraj Garu, for his valuable message and support for this particular FDP program. I thank Dr. S. Madhusudan Rao sir for his support for the entire FDP program. I also thank our honorary chairman, Dr. Nana Radhakrishnamoorthy Garu, for his constant support. I thank Principal Govindarajan sir, Vice Principal Sunita, Dr. Sunita, and our OSD and Foundry Director, Sri P. Sitaramaya Garu, for their constant support. I also thank the management of Hindu College of Pharmacy for the support. I thank today's speaker, Dr. Vadivelan, for his nice speech on guidelines on preclinical research. I extend my thanks to all the faculty of Hindu College of Pharmacy and Department of Pharmacology faculty, technical and technical, non-teaching, media, all others. Once again, I thank all the participants for registering this particular FDP program. I hope this particular FDP will be useful to them. Then coming to the tomorrow program, tomorrow session will be starts at 3 p.m. afternoon 3 p.m. So tomorrow's speaker is the Dr. Anand Vijay Kumar from JSS College of Pharmacy Ooty. So that I request all the participants to join for the tomorrow's program by 3 p.m. Thank you. Thank you, one and all. Over to Pratyusha.